Dear viewers, greetings. In this video, we are going to see about absorption spectroscopy. This video contains the following topics. First, an introduction about absorption spectroscopy, followed by principle of absorption spectroscopy, types of absorption spectroscopy, components of absorption spectroscopy, steps of absorption spectroscopy, applications of absorption spectroscopy, and finally advantages and limitations of absorption spectroscopy. Absorption spectroscopy. Absorption spectroscopy is a spectroscopic technique that measures the frequency of wavelength of absorbed light as a result of the interaction between the light and a sample. Absorption spectroscopy technique provides the valuable information about the sample's chemical composition, concentration, and molecular structure. Principle of absorption spectroscopy. The fundamental principle of absorption spectroscopy is based on the interaction between the light and matter. When a beam of electromagnetic radiation passes through a sample, certain wavelengths are absorbed, resulting in a spectrum that displays how much light is absorbed at each wavelength. This absorption leads to the formation of an absorption spectrum characterized by dark lines or bands which are corresponding to the wavelengths of light that have been absorbed. Types of absorption spectroscopy. There are three types of absorption spectroscopy. The first type is UV visible absorption spectroscopy. The second type is infrared or IR absorption spectroscopy. And the third type is atomic absorption spectroscopy or AAS. The first type is UV visible absorption spectroscopy. This type of spectroscopy measures the absorbance in the ultraviolet and visible regions of the electromagnetic spectrum, typically ranging from 200 nanometer to 800 nanometer. It is widely used for studying organic compounds, biological molecules, and inorganic ions. The second type is IR absorption spectroscopy or infrared absorption spectroscopy. This type of spectroscopy measures the absorbance in the infrared region, and it is used to identify functional groups and molecular vibrations in organic compounds. The third type is atomic absorption spectroscopy or AAS. This spectroscopy measures absorbance of light by atoms in the gaseous state. It is used for trace element analysis in various samples such as metals in environmental and biological samples. Components of absorption spectroscopy. There are 5 components in absorption spectroscopy. They are light source, monochromator, sample holder or cuvette, detector, and finally data processor. Components of absorption spectroscopy. The first component is light source. Light source provides a continuous spectrum of light, and common sources of light source include tungsten lamps for visible light and deuterium lamp for UV light. The second component is monochromator. The monochromator selects the specific wavelength of light from the light source. It typically uses a prism or diffraction grating to isolate the desired wavelength. The third component is sample holder or cuvette. A transparent container is called as cuvette. It holds the sample and the light passes through the sample and its absorbance is measured. The fourth component is detector. Detector measures the intensity of the transmitted light after it has passed through the sample. Common detectors includes photodiodes and photomultiplier tubes. The fifth and final component is data processor. Data processor analyzes and displays the absorbance data, often as an absorption spectrum or as a concentration measurement. Steps of absorption spectroscopy. The first step is preparation of sample. Prepare the sample to be analyzed, which could be a liquid, gas or solid. For liquids, it is often placed in a cuvette. The second step is selection of wavelength range. Choose the range of wavelengths or frequencies of electromagnetic radiation to be used for analysis. This depends on the specific absorption characteristics of the sample. The third step is exposure to light source. Direct a beam of electromagnetic radiation through the sample. The light source could be a lamp or a laser depending on the wavelength range. The fourth step is transmission through the sample. As the light passes through the sample, certain wavelengths are absorbed by the sample while others continue through. The fifth step is detection of transmitted light. The transmitted light measures the intensity of light that exits the sample using a detector. This can be photodetector or a spectrometer depending on the type of spectroscopy. The sixth step is data acquisition. Record the intensity of the transmitted light as a function of wavelength or frequency. This data is used to generate a spectrum. The seventh step is spectrum analysis. Analyze the absorption spectrum, which is a plot of the absorbance or transmission which is acted against the wavelength. Peaks or dips in the spectrum indicate the specific wavelengths absorbed by the sample. The eighth and final step is interpretation of results. Use the absorption spectrum to identify the sample or determine its concentration. Compare the spectrum with known reference spectra or use it to deduce the presence of specific functional groups or compounds. Applications of absorption spectroscopy. In chemical analysis, the absorption spectroscopy quantifies the concentration of chemical substances in solution and identifies compounds based on their absorption spectra. In pharmaceutical industry, the absorption spectroscopy ensures the correct concentration and purity of drugs and active ingredients. In environmental monitoring, the absorption spectroscopy detects and quantifies the pollutants in air, water, and soil. In biological and medical research, the absorption spectroscopy analyzes biomolecules like proteins and nucleic acids and study the enzyme kinetics. And in material science, the absorption spectroscopy characterizes the materials and evaluates the properties of coating and thin films. In food and beverage industry, absorption spectroscopy monitors quality and concentration of additives, colorants, and nutrients. In clinical diagnostics, the absorption spectroscopy measures the biomarkers and analytes in blood, urine, and other biological samples. In quantitative analysis, this absorption spectroscopy is very helpful in determining the concentration of a substance in a solution, and in qualitative analysis, it is very helpful for identifying the substances based on their unique absorption spectra. Finally, in kinetic studies, absorption spectroscopy is very helpful for measuring the rate of chemical reactions. Advantages of absorption spectroscopy. The absorption spectroscopy offers several advantages that make it a valuable technique in various fields of research and analysis. Here they are the key benefits. First, wide range of applications. It can be used for both organic and inorganic compounds in fields like chemistry, biology, environmental science, and material science. Next, the qualitative and quantitative analysis. In qualitative, by applying Beer-Lambert's law, the concentration of substance in a sample can be determined with high accuracy. In qualitative analysis, different compounds have unique absorption spectra and making it possible to identify the unknown substances based on their spectral fingerprints. Next advantage is non-destructive technique. This is crucial when analyzing the valuable or limited samples. And high sensitivity. This technique is highly sensitive and can detect even trace amounts of analytes. The next advantage is fast and efficient. Measurements can be conducted rapidly, often in just few seconds to minutes. Minimal sample preparation. Sample preparation is usually straightforward, especially for liquid samples. This reduces the time and complexity of sample handling and making the technique highly convenient. Next, the versatility. The absorption spectroscopy can be applied across a broad range of wavelengths. Cost effective. The equipment used for many forms of absorption spectroscopy is relatively affordable when compared to the sophisticated analytical techniques like mass spectrometry or NMR. Next, the real-time monitoring. The absorption spectroscopy can be used for real-time monitoring of chemical reactions, changes in molecular states or environmental conditions by continuously tracking the absorbance changes over time. Next, in situ and remote sensing applications. This can be applied for in situ analysis as well as the remote sensing due to its ability to detect absorption signatures from a distance. The next, the compatibility with automation. Absorption spectroscopy is easily automatable and allowing the integration of other analytical tools or systems for routine analysis in industries or research laboratories. Next, the wide dynamic range. The absorption spectroscopy has a broad dynamic range. It means it can be measured a wide range of concentration from a very dilute to reliably concentrated samples without needing the extensive sample dilution or adjustments. Finally, molecular structure and interaction studies. This technique very helpful in studying the molecule structures, electronic transitions, and intermolecular interactions, particularly in the field like organic chemistry and biochemistry. These advantages of absorption spectroscopy contribute to a widespread adoption of this spectroscopy in laboratories and industries and enhancing its role in analytical chemistry and other scientific disciplines. Limitations of absorption spectroscopy. Absorption spectroscopy has several limitations despite its advantages. Understanding these limitations is important for effectively applying the technique. First, the sensitive to interfering substances, limited to absorbing species, complex sample matrices, path light variability, non-linear relationship. It requires calibration, limited detection range, sample preparation issues, spectral overlaps and overlapping peaks, sensitive to environmental conditions, equipment limitations, and finally, resolution constraints. Despite these limitations, absorption spectroscopy remains a widely used and valuable technique due to its ability to provide detailed and accurate information about the concentration and properties of absorbing species in a wide variety of samples. Dear viewers, that's all about the absorption spectroscopy. Thank you for your support. Thank you.